Right, everybody, we'll get you to take your seat. I'm getting the nod that the majority of you are in the room, so to keep us moving, we'll start the next session. So very similar to what we had in the last session, we've got 4 amazing speakers. Again, very quick talks, so pay attention, and then we'll have a round of questions at the very end. So really great pleasure to invite the following people to stage. We've got Suzanne Rowe from AgResearch. We've got Lorna McNaughton from LIC. We've got Jason Wargent from Biolumic, and we've got Linda Johnson from AgResearch. And Suzanne is going to kick her off, but kick us off. We had a good laugh about me taking my boot off and throwing at her if she didn't leave the stage on time, which Suzanne is not known to be on time, but today it's going to be amazing. Right, Suzanne? Over to you.
Thanks for that lovely introduction, Sinead.
You're welcome.
I'm watching for the shoe. I'm almost tempted to just wait for the shoe, because who wouldn't? Okay, so I'm going to talk about breeding. We've heard a bit about breeding today. We've heard that it's an established technology. So I'm going to talk about some work that we've been doing to try and make it go faster, because we always want to get better. I'm not going to talk about the other technologies, because I think we've talked about that a lot. But I am going to talk a little bit about why we think that breeding is a good mitigation solution and a good tool in our toolbox. So breeding's available now. The systems are in place. We can rank animals and we can get a breeding value and we can choose a parent for the next generation. And one of the things that we've seen over the last probably around 10 years as we've gotten out into industry and measured something like 15,000 animals is that there are lows in every single flock or herd that we approach. So that tells us that it is available now and that we can select animals within the environment that they are established in. It's a one-off expense when you buy a parent and you breed that parent, particularly with males. If you buy a ram, you breed it to, 150 ewes, then those genetics are there. They're low methane genetics, and they're there in perpetuity. It's very low cost. John and I did some modelling a few years ago, and we looked at the cost of mitigating 4.5 megatons between now and 2040. And it came out around $2 a tonne. So Tom's $50 a tonne, works really well in this model. But it is slower. We do get slower incremental change, and we have been challenged by that. We have been challenged by, well, the other mitigation strategies actually might make a bigger difference, and they might be quicker. So could we go faster? So I just want to talk a little bit about demonstration. We have a standard flock. A genomics flock at AgResearch, and John's been recording for about 40 years. It's under standard environmental conditions. My lambs look just as bad as everybody else's down in Southland at the moment. But we've been tracking it, and in 2017 we implemented selection for methane at $100 a tonne of an economic weight alongside all of the other traits. And I just wanted to show you that basically implementing selection for methane has lowered methane by 1% a year. It's got quite a strong economic weight. It hasn't slowed growth, so we're still getting productivity. We've talked about productivity gains. And when we reach out and we take out the learnings from 2017 to today to 2050, we are seeing a potential for a 27% reduction while still seeing $20 per gross margin per ewe added. So we know it works, and we've shown it works, and we continue to show it works. So the point of the talk, we know we can measure it. We think we understand some of the biological basis. How can we predict at scale? How can we scale this into something that's useful not only in sheep, but in other ruminants, and particularly in cattle? So Peter told us that it's not really that simple, but we do understand a little bit about the biology. We know that this animal is eating feed, and the microbes are breaking down that feed, and as methane is being lowered by breeding for low methane animals, we're changing that microbiome. So we don't know if we're changing the environment or how we're changing the environment. We just know that the low methane animal has a different microbiome. We also know that that microbiome leads to a different fermentation of feed. And we know that the outflow from the rumen, or the sort of carb source, the fatty acids that are feeding the animal, are different in the low methane animal. And we know that that gets passed on down the chain, and that because it's a different carb source, that actually what we see is a different source of, or a different fatty acid profile in the meat and the milk. So we've got clear biological indicators of changes in the system that are associated with low methane. So can we work backwards? Can we use some of these indicators, which are much easier to capture and measure, to find these low methane animals without having to carry out very expensive measures on millions of animals? So we have targets. We have methane, and we have rapid methane. We had this idea that actually we can measure methane very, very, very quickly. Sorry, I'm going to go back. So we've got methane, we've got rapid methane, we've got the animal's DNA. We can just use prediction from genetics. If we know we've got low methane animals, perhaps we can just predict using the animal's DNA. We can use the microbes. We can use the rumen outflow itself, and we can use the composition of the meat and the milk. So those were the tasks that we were set, really. Can we find biological proxies that have utility and can be used at scale? And one of the things that we've done is, as I say, just develop rapid ways to capture methane in a short-term trial, which basically means that we can assess other technologies. We have to be really careful with some of these technologies, though. We have these portable chambers that accumulate methane. So we know that we can put a sheep or a cattle beast in, methane will accumulate, and we'll be able to measure that, and we'll be able to rank these animals. This is done under a specific and set protocol, and it doesn't tell us about the absolute methane that the animal is producing over the day. So it's an alternative measure that's really, really useful for breeding. It's not a miracle, but it does rank animal well. It is fast and it is low cost. So it is a really useful tool that we have. Microbiomes for us were the sort of mecca. That's where we really put a lot of effort. Microbiomes is where everything is happening. They're digesting the food. They're producing the substrate for the archaea. That's where the methane is coming out. So we've spent probably around 6 or 7 years working out how to sample an animal's microbiome, where to sample an animal's microbiome, what to do in the field if you don't have dry ice. We've ordered ice from party supplies and done all sorts. Developing preservatives, being able to capture a small amount of rumen fluid, bring it back to the lab, sequence it in a way that's meaningful, predict methane and rank animals, and finally move on from that in terms of if we are going to sample animals using a stomach tube, can we sample animals using an oral sampling device? So that's a swab. So can we get out there with a swab, wipe it around an animal, find what's the bugs that are active in the rumen? And it sounds like, it sounds difficult, and it has been difficult, but it's surprisingly predictive. And finally, the fatty acids. Can we take the fatty acid composition of milk or meat and see if it tells us whether an animal is high or low methane? So we did some herd testing on our low methane sheep and our high methane sheep, and we used LIC, and we sent samples away for herd testing, and they came back and the fats in the samples were exactly the same. So you couldn't tell the difference between highs and lows. But when you drilled down and looked at the fatty acid composition, they were very, very different. And when we went back to LIC, who've got some really great experts in this field, in particular help from Catherine Sanders, and got their spectral profiles that they'd used for our samples, then we could actually find signatures in those spectral profiles that gave us a signal that basically differentiated those fatty acids. And this is work that Timothy Bilton's been doing. I won't talk about it. There's a poster out in the foray. Timothy's worked really hard on this for a number of years. He's a very talented statistician, and we certainly wouldn't be where we are without his work. But what we ended up with is the genetic signal for each of the proxies and how they all relate to each other. I'm obviously not going to show you all those numbers today. But what I do want to show you is that all of the proxies that we've looked at were moderately heritable, apart from the one coming out of the rumen. that sort of carb source, those fatty acids coming out of the room and we can't capture. We're probably not doing it at the right time and we probably don't have the right protocol. So we did a poor job of that, but the rest were really moderately heritable and they're all highly genetically correlated with each other. And that means we could use any one of them in a breeding program to breed for the other. So that means that if I don't measure methane, but I measure the microbiome, I can go 80% as fast just using the microbiome. So I can get 80% of my genetic gain by measuring the microbes with a tube rather than using a chamber. And in truth, there will need to be a combination and there will need to be calibration. But really excitingly for the dairy industry, the milk test got me 70% of the way. And when we think about that in terms of what the money that we spent, and Sinead, this is my last slide, you can keep your shoe on. If we look at the money that we spent in order to get a breeding scheme running, so let's say we bought one respiration chamber measure. For that measure, we could get 5 rapid measures, 25 fatty acids, 50 microbial profiles, or 1,000 herd tests. Now when we look at that, what that means in terms of genetic gain, I've set my genetic gain, so the rate at which I'm going to improve methane at the rapid measure, because that's sensible for a breeding scheme. I can probably go half as fast if I use respiration chambers, but I can go twice as fast if I use milk tests, and they're really easy to catch, and they're being collected anyway. So there we're starting to move towards biological proxies that tell us about productivity, enable us to select for low methane, and can be carried out at scale. Next steps for us are looking at this in cattle, looking at it in beef, looking at it in dairy animals, getting it out at scale and really putting our money where our mouth is and getting the job done. So I will basically leave it there.
Suzanne. I thought you did say you were going to test me and whether I would take my shoe off and throw it to you, and I was looking, maybe I might have to, but thankfully you finished in time. Lorna, over to you, please. Thank you. Yes, I am Irish. Number 2 in the rugby team, just to inflame a few people. New Zealand is number 3.
So good morning, everyone. It's my pleasure to be up here with an update on the program that CRV and LIC have been running on breeding low-emission dairy cattle, very much following in the great footsteps that Suzanne and John have put for us to follow. So I thought we'd start by thinking about where we've been in our program. So genetics does take quite a long time. So back in 2021, we really kicked off for real with our first bull measurements. So we measured about 300 bulls in that year, and every year since we've been measuring young bulls at LIC and CRV. So we've now got over 1,100 bulls in our data set. But as Peter Gluckman mentioned, physiological state is likely to have an impact on methane emissions. So we need to make sure that by measuring bulls that we see those differences in the daughters. So in 2022, we kicked off a trial to examine that. And again, cattle have quite a long breeding period, so it takes a while. So we selected the top and bottom 25 bulls at that point. We'd measured about 500 animals by that stage and inseminated them into a large herd down at Pamu. Those daughters were born in 2023, and then just this past year, finishing late September, we've measured 393 of them. There were, I think, 5 little madams that wouldn't put their heads in the green feed, so we don't have any data for those ones. Then our kind of next phase of the program is the lactating cow measurements. So that's measuring those same daughters who we've just captured their methane emissions as lactating cattle, making sure that we still see those differences. And then, all going well, hopefully releasing bull breeding values in late 2026. So most likely in time for the 2027 breeding period. So just a refresher on how we actually do the measuring. So we use a green feed, and it's very handy that Christian has brought one along. So if you want to go and get in depth about how it works, he might even give you a mint if you put your head in and breathe into it. But you can go out and have a look out there. So we measure methane through the green feed, we measure feed intake, and that means the animals need to come into a barn. And that's because some of that variation in methane is about how much they eat, and at this stage of our program, we want to make sure that we're capturing feed intake, so we're not selecting for low methane through animals that aren't eating a lot. We weigh the animals 3 times a week while they're in, and they're in for between 35 and 40 days. So we collect a rumen fluid sample in there, and we collect lots of variations of that rumen fluid sample, including the swabs that Suzanne alluded to, and that data all goes down to Suzanne to support this proxy development program, because we are very keen to get a whole lot more data as well. So it's very exciting. We've finally got our heifer results. Olivia's been working incredibly hard on getting the data analysed. It takes a while for it to come back from the methane measuring people CLOC, and we have got a difference in the methane emissions between the daughters of the high and low methane bulls, which is super exciting. This first slide has got the total methane emissions, so that's the grams of methane per day, and it's also got feed intake. What we actually selected the animals on was methane adjusted for feed intake. So on the next slide, I've got some more results around that. But there is that small difference, about 5 grams per day total between the lines. And you can see there that the feed intake is not different between the 2 lines. And that was what we were really looking for with the selection that we were doing. So then methane yield, so that's grams of methane per kilogram of dry matter eaten. And again, you can see the clear daylight between the lines. It is significantly different. It's about 0.8 of a gram for every kilogram of feed eaten, and it's dangerously close to the 0.7 that we were predicting we might get based on the breeding values. And then if you have a look at the breeding values, so all the data we collected went across to Helical, who are an independent third party, and they've produced breeding values for these heifers. And again, you can see that they differ between the highs and the lows. So that's a big tick to our program continuing, so we're all very relieved and happy about that. So then, time to think about kind of where we're at. So in our bull work, we've identified that there is genetic variation in methane, and we've now ticked off that the young daughters, at least, of high and low methane sires are themselves either high or low in methane on average. And that was a pretty good finding, given that we'd only measured 500 bulls and we were only selecting on the male side. But the next stage is thinking about that lactating animal. So does measuring a young growing heifer give us a good prediction of a lactating cow? We really hope that it does, because it's a whole lot easier to do those measurements on young animals. They're much smaller, they eat less, it's a whole lot cheaper to do the research. And then the next stage, what are the genetic correlations between methane and other traits? So in the literature, it's a bit conflicted, but there's a really nice big study just come out of the Netherlands where they have measured thousands and thousands of animals, and the genetic correlations between milk and methane are looking kind of low, so less than 0.2 and mostly a lot closer to 0 than that. Interestingly, the highest correlation was with fat percentage, which may line up with some of those fat stories that we're thinking about, hearing about from Suzanne. So my final slide, just to think about where we are. So really positive progress. We're working along the pathway as we had hoped to be, but we've still got a couple of big pieces of work to do, and that's that lactating animal trial and gathering enough data for those genetic correlations. We're on track at this point for breeding values in 2026. And the really nice thing about genetics is it's verifiable, so we can track an animal's parentage LIC and CRB have really good records of which cows get mated to which bulls, and we can use DNA to verify that that is the case, so we can do breeding values through the population. And there's a really clear adoption pathway. So the farmers just need to keep buying the semen and selecting the right semen, and then we can get it out to the industry very quickly. And just before I finish up, I'd like to say thank you to particularly the on-farm team who do a lot of the hard work. I don't spend much time out there these days, and the massive kind of infrastructure sitting behind that. Thank you.
Thanks, Lorna. Exciting stuff, and you heard it here first. So really nice result. Yeah, over to you, Jason.
Okay, tēnā koutou katoa. It's a great pleasure to be here. It's my first time at the conference, actually, but it's been a really excellent experience. I'm going to just provide a quick overview in the next few minutes to who Biolumic is, what we're doing, and also what we're doing in a new space for us, which is providing another type of opportunity to be able to access low emissions pasture. So at Biolumic, we program powerful crop traits by applying treatments of light to seed, UV light. We're a company that spun out of Massey in 2013. We have a headcount of over 30 now. We're distributed across New Zealand and also in the United States, where we've been doing a lot of work in crops like corn and soybean, as you might expect if you're working on seed traits. The mission for the company is really around the challenge that exists for crop breeders and others that are involved in procuring traits in agriculture. We know that traits are badly needed, traits such as increased yield, increased composition, whatever they might be. We have a paucity of traits, and we have challenges in being able to bring traits to market. These are just a few of those challenges. Bringing traits to market can be very slow, depending on the nature of the technology that's producing those traits in different varieties of crops. They can be enormously expensive. They can be encumbered by significant regulatory resistance, depending on the territory and the crop and the trait that we're talking about. And also limited trait power. We believe strongly that plants are packed with enormous potential for trait access using current tools we have and future tools that we have. And we believe our tool is one which could help us to be able to access all kinds of different traits and also be able to stack traits. A common roadblock is not being able to have more than 2 or 3 traits of value within one package of one variety. And we hope that our technology can try and bring other ways of trying to solve that challenge as well. So our solution is relying on these hundreds of millions of years of plant signaling that have already evolved. Plants have far greater capability today than humans have currently been able to access through the science that we have to date. So we treat the programming of plant signaling like a computer program. We know that light treatments applied to a seed, which I can say more about in a second, are able to change the way that a plant develops. So we apply our treatments using different approaches that I'll talk about, and then you've essentially imprinted that seed, and then you've imprinted that crop so that when it is sown and starts to grow, you have permanent changes in the developmental plan for that crop, and you access those traits accordingly. Just in terms of the practicalities, so currently we're applying treatments of light to seeds. We're using approaches like single layer conveyance. We can apply the light directly to the seed. It's a quick process. One of the quickest treatments we've used so far is seconds, and other treatments are taking mere minutes. It's a dry process. There's no material added to the seed or the germplasm. We're not taking up that valuable real estate around the seed surface. It's a very quick process, relatively speaking, compared to a lot of current trait and breeding programs that exist. We know we can stack traits, so we look at traits such as increased yield in corn and increased establishment and others. We also know that we can call what we say locking in traits as well. So we've had some really exciting results in the US this season, where last season we worked on inbred parent corn seed, treated the parents. This year, we've harvested hybrid corn yield trials where those untreated hybrids have had enormous increases in yield. And we believe we've found a way to leverage our technology's action by going through the hybrid vigor process. Our technology is not GM, but we're entirely complementary to GM approaches. Working in crops like corn, we've been embedded in varieties that are GM for years already. And it could be a way, we don't see ourselves as a silver bullet, to be able to leverage other approaches like GM by applying our technology as well. We can apply treatments, we call them light recipes, rapidly to any crop variety. If we've already developed on a trait in a particular crop, we have a very quick screening process to put our light treatments or recipes against a new variety that a seed developer wants to launch. And we've been spending a lot of time on understanding proprietary novel mode of action that's actually underneath our technology, which obviously can help us exploit the opportunities even more. I won't say too much about this today, given the time we have, but the way in which we map these treatments to the varieties and to the traits is using molecular biology techniques. We're essentially using marker-based gene expression modeling. So we target certain genes, or we map certain genes that are involved in our processes, and then we use screening and models to be able to take one of what are, we believe, billions of these light treatment regimens that exist for us, billions of UV light treatments. We can cut through those by using modeling approaches to choose the recipe that can give us the trait or the stack traits that we're looking for. So just a little bit on the commercials, then we'll talk about ryegrass quickly. We're already commercially engaged. We've been in market in the US with corn approaches. We have a seed treatment machine, hopefully on a ship somewhere in the Pacific at the moment, on its way to the States to deliver our first machine, and that's going to be treating inbred corn seed in Q1 next year. Our new program in ryegrass, we had an opportunity to start to bring exciting gains in the company's technology back to New Zealand for New Zealand benefit, where we were born and where we live. Focused on ryegrass. The first target is increasing lipids. I think we've had plenty said about that mechanism opportunity there, or mechanisms to increase lipids and to reduce methane, and that's what the company's currently targeting in annual ryegrass, perennial ryegrass, and we're looking to expand that work with some near-in partnerships as well. At the moment, this work is being supported through AgriZero as an investment partner. So where are we? What are we doing next? What's the plan? The chart on the left is actually an early indication of the work we started seeing quickly in the program, whereby increasing the lipid concentration of ryegrass, there's that connection, which we will test on farm, of course, to be able to reduce methane in Biolumic treated grass over persistence periods to show that we don't just get a temporary effect, but we get an enduring effect in that grass profile. That was a starting point. We've already started to see the lipid target that we're after, around 6% of dry matter concentration. We've seen signs that we're already able to reach those levels. That will be baked in with our markers, our gene markers. We expect to be in the field by next winter in annuals, for example, and then we're going to be looking to get into longer-term field trials and obviously animal trials to prove out the product claims accordingly. And then just lastly, the graph on the right is a postscript really to what we're talking about. We are engaged on the lipid target already through the support of AgriZero, but we already know that we can increase or trigger other traits in ryegrass. There's been a lot of talk this week about the proposition for the farmer. Can you provide productivity? And we believe we can. We've seen gains in dry matter productivity in an equivalent per hectare basis. We've seen increases in metabolizable energy and other traits like nutrient use efficiency. So our enhanced program that we're looking to put together soon would allow us to look at this soil to grass to animal to milk to outcomes view of what the full impact of our stacked traits could be in a pasture system while reducing all sorts of emissions accordingly. And with that, I'll wrap it up there. Thank you very much.
And Linda is going to conclude our presentation. So over to you, Linda.
Good morning, everyone. I'm Linda Johnson, the Science Group Manager for Resilient Agriculture. I just thought I'd very briefly tell you what the Science Group does. So we are developing numerous innovative technologies to help contribute towards a low emissions future on farm with improved environment sustainability outcomes and resilience to climate change. So there are 9 or 8 science teams actually with expertise across plant science, plant microbiology, rumen microbiology, ruminant nutrition and physiology, as well as significant expertise in measuring methane. So, HME, today I have the great pleasure to talk to you on one of our plant genetic technology programs, HME perennial ryegrass. We have 2 other genetic technologies which we hope that HME will be stackable with in the future. So HME, what does it stand for? High metabolizable energy. And this leads me to the concept of the program, which is to elevate the lipids or the fats, as well as the gross energy of ryegrass. This is to improve animal productivity, animal nutrient and animal productivity, and ultimately as well to produce a low emission solution for on-farm. And it's a pastoral-based solution. It does use genetic technologies. So genetic modification is used to increase the oils in the leaves. And we do this through introducing 2 genes. One gene is called DGAT. It's a modified universal enzyme, and the role of that gene is to synthesize oils to a high level. But I think the real feature of the technology is, in fact, the oleosin. So a modified gene produces this protein called oleosin, and what it does is it actually stabilizes the fat or the lipid. And if you can have a look in this big oil body here, you can see thousands of droplets, which would otherwise just coalesce to one huge, large globule. And so these oleosins have shown us that even in field trialling, we've been able to see the stability of the change. So we've been able to see a 2-point difference or a doubling of fat in the leaves, which is fantastic. So this program is designed to boost animal productivity while also reducing our greenhouse gas emissions. But why are we interested in ryegrass? Well, as most of you, I'm sure, are aware that New Zealand's, the backbone of New Zealand's feed base is based on not only ryegrass, but ryegrass with its associated endophytes and white clover, and they are the most cost-effective and widely used feed. Not only that, but this technology is scalable, and it can be incorporated just into seed mixes sold with the companion species white clover and along with the associated endophytes. So what are the associated sector benefits of HME technology? So first of all, with our 2-point difference in the elevation of leaf lipids, we are estimating a reduction of about 8 to 10% in methane. This is based on a number of studies. So you've heard earlier today, there's a study by Granger and Buchanan, as well as by Moate, that have showed, based on a 1% increase in dietary fat, this could correspond to 3.5 or 5% decrease in methane emissions. So that's where we get our estimation of about, well, 7 to 10%. As they say, the proof will be in the pudding, and we hope that will be a Christmas present for us based on an animal trial. Not only are we seeing reduction in in methane, but also in the other greenhouse gas, nitrous oxide. And we are conservatively estimating this to be about 10%, but it is also based on an experimental study. So we've done a greenhouse study using mesocosms where we've sown HME as well as the control and compared and added urine, and the outcome is that we've seen a reduction in nitrous oxide in soil grown with those plants by about 10% and even up to 30%, but that's really dependent on the trait that's been introgressed into those plants. So obviously this is called high metabolizable energy, so it won't surprise you that we're expecting an increase in the metabolizable energy. And in fact, we have done some experiments where we've measured the metabolizable energy, and we've done some projections where we expect to get up to 10%. For those farmers out there, this is approximately an additional 1 megajoule per kilogram of dry matter. So along with this increase in metabolizable energy, we expect to get an increase in animal productivity of about 10%. So as I've mentioned, it's stackable with clover and endophytes, and these could be GM or non-GM technologies. And with the regulatory landscape changing, we're really hoping that this technology can be adopted by New Zealand farmers. So here I present the estimate for the incremental farm gate value of HME technology for New Zealand and Australia, which has been based on modelling carried out by independent consultant for us. So here we're looking at a period of over 20 years, and it's based on conservative adoption rates, which may go up to approximately 80% at least for dairy, with a renewal rates ranging of 8% for dairy and a lower 2.5 to 4% for sheep and beef. What you can see there is over those 20 years, the incremental benefits that have added up cumulatively over those would be $24 billion, which is significant. Or another way of looking at it, if you just looked at the New Zealand dairy sector, for example, where we have modelled an adoption rate of 80% from year 15, we're seeing an additional $1 billion per annum. So embracing these plant genetic technologies, we think, will create a bright future for New Zealand by achieving improved environment and sustainable outcomes for our farms and resilience to climate change. So the numerous benefits of HME and our other two programs, which you can see the details here, so they are the high condensed tannins white clover and also gene-edited endophytes, which also in the future could be used to express methane inhibitors in plantar. These could be stacked, and of course we don't know whether or not these stacked would enable benefits or not, are outlined here, and we think that they will be great pastoral-based solutions for improving emissions on farm, reducing inputs, improving animal health, and increasing productivity of the whole system from the forage all the way to the animal. It's really been an exceptional effort to get where we are today. And we anticipate at least another 4 to 5 years of science. It has already taken us 20 years. It's been a really difficult program, but we are making amazing progress, and potentially another 2 to 5 years for commercialisation, where breeding companies will take up and use plant breeding to integrate this trait into elite germplasm. So with that, I would like to close up with my acknowledgements. In particular, I need to acknowledge the HME ryegrass team, particularly the brains of this, the principal scientist Nick Roberts, as well as Richard Scott, our nitrous oxide team, and our funders. And with that, I'd like to thank you very much for listening.
Thanks, Linda, and thanks to all our speakers for keeping on time. And because we've been able to ask questions to the previous group, we're going to focus the questions now on these 4 individuals. And there is quite a lot of questions, guys, so brace yourselves. So I'll start off with you, Suzanne. Someone's asked a question around, and others please join in, about the need to understand stackability of approaches. And this individual was wondering whether you'd got any more information on how the low GHG sheep respond to other mitigations, maybe such as an inhibitor or other pastures. Got any comments on that?
Yeah, great question. I'm desperate to look and see how the low methane sheep stack up with all of the other technologies, and we're certainly working towards a program where we do that. They're a resource that's unrivaled. We've done a little bit of sort of feeding things like brassicas, and we definitely see differences in the way the lines react. So yeah, I'm really hoping that we can talk to the people in this room, decide on the best technologies to stack, and put together a program that's informative and useful.
Yeah. Suzanne, another question that's come in for you is, does the different fatty acid composition in low methane animals have any impact on the nutritional value of the meat and milk? I think that's important for the processors.
So at the moment, what we see is lower short-chain fatty acids, more branched-chain fatty acids, and more what we call conjugated linoleic acids. So from a human health perspective, it's a really nice story. I think it's nuanced. I think if you were making a particular type of cheese, you want to know exactly what was going on, and you be careful to acknowledge that. But in general, it looks as though the profiles are good in terms of the meat chain and the food chain.
Cool, thanks. Lorna, Question here on what breed are the bulls? Are they predominantly dairy breeds, or are there some beef crosses being looked at?
Yep. So at this point, they're all dairy bulls, so a mixture of Friesians, Jerseys, and Kiwi crosses. But the beef project that is getting up and running will no doubt be looking at beef bulls.
Yeah. Also, another question for you: have you compared feed conversion efficiency with methane emissions?
So we have not. We've been collecting that live weight data so we can, and we have a PhD student who hopefully her second piece of work will be doing that.
Oh. And this is for Suzanne and Lorna. Have you modelled adding other ways for the genetics to go faster? Example, combining mixed sex semen and short gestation, improve fertility. Any thoughts on that question?
We've got a student looking at it at the moment, but the answer is largely no. But absolutely right. You know, reproductive technologies may well play a role, and we're starting to delve into it. But actually, that's your bag.
Yeah, so we haven't done any specific modelling, but we already obviously do a lot of reproductive technologies in our breeding program. So a lot of ET, there's increasing sex semen usage, and particularly in our breeding herds. Cool. So we would be looking obviously to maximise that once we're very confident that the genetics are ready to be released.
Thanks, Lorna. Jason, question here: what levels of methane reduction are you expecting from treated ryegrass?
Yeah, so this gives me a chance to say all the things I didn't have time to say earlier. Thank you. We're targeting a 2 to 3% increase in lipid concentration. So that would indicate, depending on your science you're leaning on, that we'd be getting a double-digit reduction in methane from that trait. So we're going for about a 6% lipid concentration of dry matter. Obviously, that does need to be fully proven in the field, as does obviously that space. More field studies are happening all the time. But we believe there could be other mechanisms that we're triggering which could add further beyond the lipid trait. And so we'll know more about that as we get into the field.
Yeah. Another question here for you is, are the trait expressions inheritable, or will each new generation of seed need to be retreated? And also, just secondary to that, given that the UV technique is non-selective, how does it tackle new variants of disease, and how does it ensure risk mitigation against unexpected consequences?
So on the first question, obviously it's relevant in ryegrass with daughter tillering and perennials and so on, and we've done a tiny bit of work on that already. We've been going on this since April, where we've started to see gene expression when you basically daughter out tillers from a existing tiller system. So that gives us some promise of what we'll eventually be studying in the field. We've also done this work in other crops, not just hybrid seed varieties, but other seed varieties, where we've seen heritability through a seed production stage of the crop's life. So we're actually very, very optimistic that we will have this follow-through of the trait remaining in place, no matter what stage is occurring after. And we're doing some work around studying possible mechanisms, epigenetic mechanisms, and other inheritance-based mechanisms as well. And then on the second question, I mean, we're studying at a molecular level what the effects of our treatments are all the time. As I was saying earlier, we're outside of a regulatory framework here. We're not ACVM. We're basically enabling natural gene expression of genes that already exist in the plant. And we've seen no evidence so far of the induction of genes that would take us down a path that we wouldn't want to see. And because we have this massive palette, these billions of recipes, we can do a lot of that work at a molecular level without needing to, go too early in terms of particular treatments going further down the commercialization path, without you knowing a lot about what's happening inside the physiology of that path. But I think our approach with UV is a lot more focused and specific in terms of gene expression to that final point in the question than anybody else would have used to date with work that's been done with UV in the past.
Thanks, Jason. Linda, is there already some insight into how a high ME ryegrass handling challenging climates, is it resilient? Is there some insight into how a high ME ryegrass may handle challenging climates? Is it resilient?
Yes, I think it is. We have done some field trials in the US, and with that we have seen that elevation, that stable elevation in fatty acids. We've also experimentally determined that improvement in gross energy, and I think when you look at those traits, with the fact that that increase in fat, that it is stable, and also even with seasonal differences, so the lipids in a season actually change, and what we've constantly seen is that difference between the HME and the control. So consistently seeing that 2% increase will mean that we reliably should be receiving a climate resilience with respect to lower emissions, which I think is really important. If you think about, I guess, adaptation to climate change and other factors such as extreme weather events, etc., that is more problematic. We can only deal with the genetics that we have with perennial ryegrass, but in our other programs, in our plant science programs that we have going on, what we found out is that the genetics that we've used in perennial ryegrass, which, you know, the driver has always been for improved plant productivity, improved yield, but now, of course, there are these climate change drivers, and so what we're doing is we've discovered that the germplasm for perennial ryegrass has been very, very narrow. And so what we found is that if you go back to germplasm that's actually stored in the Margot Ford Germplasm Centre at Grasslands at AgResearch, you can actually integrate those more diverse traits when you're particularly looking for example, drought tolerance, improved nutrient availability. And with our technology, the HME technology, what we will be doing is that seed companies will be taking that and integrating that with their elite cultivars, and they will be taking our pre-breeding, where we've been actually looking for these important traits of improved nutrient availability, drought tolerance, and those should be able to still be integrated into that HME ryegrass, which has that specific trait. So yes, I think in the future we can look forward to perennial ryegrass still being around for a long time.
Yeah. Another question, and it's a bit similar to what was asked to Suzanne. Does the HME ryegrass change the fatty acid composition in the milk? And does this change the production yields for dairy processors producing fat-based products like cheddar and butter? Any thoughts there, Linda? Linda's like, negative? It's for you, Linda. Sorry, I'll go back. It was around HME ryegrass changing the fatty acid composition in the milk. Yes, and does this change the production yields for dairy processors producing fat-based products like cheddar and butter?
That's a really good question. I don't have the answer for that. What we do know is that we haven't gotten to that point of doing an animal trial, and yes, that would need to be done, but we have analysed the changes in carbohydrates in perennial ryegrass, and there is obviously the change in lipids, and there's also some other changes in carbohydrates. We are only doubling the amount of lipids, and we're not doing anything else really to the grass, so I would expect there to be no significant impact, and we're not expecting.
Okay.
Yeah.
Cool, thanks, Linda. So I think I'll do one more question, and it's the top-rated question, so it's a little bit pointy. It feels like the commercial parties are more positive than the scientists. How are we going to ensure that there will be economically viable solutions for farmers versus commercial investor returns? Who wants to put their hand up to take that question?
I'm excited.
Oh, go, Suzanne. Off you go. I know, Jason.
Yeah, I'll speak. I mean, we're a venture-backed company, and so we have investors, and we have arrangements with those investors and so on. But the bottom line is that, go back to what I said earlier about traits. We need every single tool in the box that exists or does not yet exist to give us traits in crops. Biolumic is massively excited about being able to do something on our home patch, let's say. We've been in the row crop world, as I was saying earlier. And so to bring that back to a complex challenge, such as the ryegrass space and the methane space, is really exciting. And the commercial side of that, we've obviously thought about this already. We're also looking to have strategic partners involved in the work we're doing as well. So we're not just taking investment dollars and and trying to make, non-conservative estimations of what we can achieve or what we can produce in terms of financial outcomes either. I think it's about real challenges and excellent science. And, some of the science of yesterday will be important tomorrow, but some of the great science we do not yet have, we still want that science tomorrow, whether it's something that somebody is doing today or not. And, the idea of a proliferation of tools, I think, is what this is all really about.
Yeah, Suzanne?
Can I just also say that, no apologies for being conservative in our outlook. We need to be. We can't make false promises, but we are excited, and there are solutions coming. Under the auspices of the centre and also of the GRA, we're working really closely with scientists overseas and international groups to bring these solutions. So they are coming, and we are working hard, and we are excited by them. So apologies if we look like we aren't.
I was going to say you're sounding positive, Suzanne. What?
Well, I just want to say that obviously we're very excited, and it's a great opportunity to be telling you guys about all these varied solutions today. But with respect to the fact that farmers aren't going to feel the benefit of these technologies, it's not true. And I think we are really thinking about it as scientists, that we need to make sure that actually there's going to be something in it for our farmers. Otherwise, we won't have farming here today. It's got to be economical, it's got to be viable. And that's why I presented the benefits to the farm gate for farmers. So you're actually going to see benefits with animal productivity. And I think that's really important. Any of our solutions that are going to be environmentally beneficial, that have a low footprint, they also need to still have those productivity gains. And, the government is asking us to double our exports, and we do need to feed the world.
Cool. And with that, I'd just like to thank all of our panel members. We've obviously gone through, from inhibitors to vaccines to sort of genetics to plants. We haven't gone through them all, but, hopefully you've got a little bit of an overview of what's happening, and you are seeing the progress in the science. So big thank you to all of our panel members.